All right, hello everybody. My name is Matt Bernstein, and today I'm going to talk to you about a recent project called the MetaSRA, which consists of normalized sample-specific metadata for the sequence read archive. And this is work that I've been working on together with my advisor, Colin Dewey, at the University of Wisconsin-Madison. So the sequence read archive, or the SRA, is this large repository of next-generation sequencing data. It's curated by the National Institutes of Health, and it contains sequence data from over 1.5 million biological samples across over 75,000 studies. These data come from scientists all around the world. It's very heterogeneous. For example, it consists of cells grown in culture. It consists of tissue samples, either from biopsies or autopsies. It contains data from cells that have undergone more involved protocols, such as cellular differentiation. And with this heterogeneity comes unprecedented opportunity to study various biomolecular processes and how they relate to a number of phenomena of interest to biologists, such as phenotype, disease, tissue, cell type. And some of the data types in the SRA include genome sequencing for studying genotype, RNA-seq for studying gene expression, ChIP-seq for studying protein-DNA interactions, among others. However, standing in the way of this opportunity is the poor structure of the metadata that makes analyses across studies very difficult. So it makes it difficult to query the data for a particular phenotype, say, of interest. It also makes it difficult to aggregate data across studies. And the reason why this metadata is poorly structured is that there's no standard that a data submitter is required to adhere to when they submit data to the SRA. And naturally, this results in many problems, including synonyms. For example, fibroblast of lung is the same thing as a lung fibroblast. There are misspellings. So here's an example where there's an E missing between the R and the S in foreskin fibroblast. And there are references to outside information. So ATCC CRL 2522 is the name of a cell line. A scientist will often label their sample with the name of a cell line as sort of a shorthand for all of the biology associated with that cell line. And an outsider, in order to learn about that sample, would have to consult some kind of external knowledge base that describes the biology of that cell line. So there is one piece of structure that is imposed on the metadata, and that's the fact that a data submitter must encode the metadata as a set of key value pairs, where a key describes a property and a value describes the property value. So here's a set of key value pairs from a real sample in the SRA. For example, a property is cell type and the value is fibroblasts. And here you can see a number of the issues that I just described. So there's that misspelling, foreskin fibroblast, the reference to outside information with the name of a cell line. A few other things to note is that there are different data types represented here. For example, there's numeric data with 72 hours post-seeding, passage 5. There are also different relationships between the keys and the values. So the property value no is negating the property lentiviral transgenes. That's sort of a different relationship than the key value pair cell type and fibroblasts. We sought to normalize or standardize all of these raw key value pairs, all this raw metadata, by encoding this metadata into a new schema, where this schema was inspired by that used in the ENCODE project for describing their metadata. And we broke this encoding task into 3 subtasks that involved first annotating samples with terms from biomedical ontologies. We extract real value properties. And third, we label each sample with a broad sample type category. So I'll go into detail for each of these tasks. First, I'll talk about how we annotated samples with terms from ontologies. So just to review, an ontology is a set of terms or entities together with relationships between those terms. And that forms a directed acyclic graph where nodes are terms, edges are relationships between those terms. Here I have a very small subgraph from the Uberon ontology that describes anatomical entities. You can see the blue edge encodes an is-a relationship between terms. The red edge encodes a part of relationship. So for example, some of the knowledge encoded in this small subgraph is that the cardiac ventricle is a cardiac chamber, which is part of the heart. And we used ontologies that describe diseases, anatomical entities, cell types, miscellaneous experimental factors, and cell lines. So there are a number of existing approaches that label metadata or tag metadata using terms from ontologies. And these existing approaches frame the task as that of named entity recognition. Named entity recognition is the term in computer science for the task of labeling or finding entities or terms in natural language text. So here's an example key value pair from the SRA. Source name, human fetal lung fibroblast, MRC5 cells, tetra BPY treated. A named entity recognition program might find the string fetal, map it to the term late embryo in the Uberon ontology that describes anatomical entities. So yeah, the highlight there is not showing up very well, but the word fetal is highlighted in red. It might find the string lung fibroblast and map it to the term fibroblast of lung in the cell ontology. It might find the string treated and map it to the term treatment in the experimental factor ontology. And we attempted these various tools that take this named entity recognition approach. And we found a number of problems, both that resulted in both false positives as well as false negatives. And to illustrate some of these problems, I'll go through a few examples. So here, take the key value pair disease none. A named entity recognition program would find the string disease and map it to the term disease. But this is wrong because the value none is negating the key. This is not a disease sample. So that's a false positive. A second example, take the key value pairs disease breast cancer, tissue blood. A named entity recognition program might find the string breast and map it to the term breast in the Uberon ontology. This we'd also consider a false positive due to the fact that the word breast is localizing the cancer. It's not localizing the tissue from which the sample was extracted, and this would be misleading, and we'd count this as a false positive as well. And then a third and final example, take the key value pair cell line, MRC5. A named entity recognition program might find the string MRC5 and map it to the term MRC5 in the Cellosaurus, so that's a cell line. And this is correct, but there are a number of false negatives here. If we know that the MRC5 cell line comes from male Caucasian, then we should map this sample to the terms male organism and Caucasian. So a named entity recognition program doesn't by default use any external knowledge bases for adding terms that you can know about the sample from what's explicitly mentioned in the metadata. And so the approach that we took is that we framed this task as more of an inference task, where we're attempting to infer the terms that describe the underlying biology of the sample, rather than just the terms that are explicitly mentioned in the metadata. We use named entity recognition towards this end, but we frame the task as an inference task. And at the heart of the solution that we have come up with is we build this graph data structure where nodes on the left-hand side represent key value pairs, which are provided as input. And these rectangular nodes on the right-hand side are the ontology terms, and those are the output. And then a path from the input to the output represents a derivation. And we developed a pipeline that slowly builds up this graph data structure using a number of heuristic procedures. And this graph data structure maintains the provenance of all the derived ontology terms, and therefore we can build a series of heuristic procedures that either filter terms or can add terms based on external knowledge bases. So just to give you a flavor of how we can utilize the graph data structure, I'll just show one such procedure. So in this procedure, what we're going to do is we're going to search the graph emanating from the key. So we build two graphs, one emanating from the key and one emanating from the value. And we find the term sex in the graph emanating from the key, and then in the graph emanating from the value. So the value is just the string F, just the letter F. Given this context, we know that F likely stands for female, whereas in another context in which the key doesn't encode for sex, F might not stand for female, and therefore we wouldn't map the term female organism to the sample. So we compared this tool, which we call the MetaSRA, to three existing tools that take the named entity recognition approach, Zuma, Sorta, and the bioportal annotator. We compared all of these tools across all 5 ontologies using error rate and recall, where error rate is the fraction of terms that we output that are wrong, and recall is the fraction of terms that we want to output that we do. And so the further left you are on the left graph, the better. The further right you are on the right graph, the better. The red dot is the MetaSRA. And we really sought to minimize error rate while maintaining competitive recall. And that's what we have achieved. OK, so I'll briefly talk about the next task, which is extracting real value properties. We encode a real value property as a tuple, which consists of the property, the numeric value, and the unit. And the unit we take from the units ontology. So for example, the key value pair age 20 years old would map to the tuple age 20 and year. I'm not going to go into depth on the methods here. It relies a lot on the graphs data structure that we construct for the previous task, but the precision that we've achieved is 0.98 and the recall 0.67. So high precision. There's a little bit work left to be done to boost recall. Okay, then I'm going to talk a little bit, and then next, the third task is labeling each sample with a sample type category. The ENCODE project labels each of their samples with a broad sample type. That is what general kind of sample it is that you're looking at. The ENCODE project uses 6 categories: tissue, stem cells, cell line, induced pluripotent stem cell line, primary cells, and in vitro differentiated cells. And we found that it was necessary to also label each sample with one of these sample type categories because there was oftentimes ambiguity into what sample it was, even when we mapped ontology terms to the sample. For example, let's say a sample is annotated with both the terms stem cell and fibroblast. There's some ambiguity as to whether the scientist started with stem cells, differentiated them into fibroblasts, or started with fibroblasts and induced them into potency, thereby deriving stem cells. And so we also label each sample with these sample type categories. We more rigorously define them by constructing this flow chart, which describes different protocols you can perform on a sample and the sample type that would result from those protocols. And since there are relatively few of these sample type categories, we used a machine learning approach for this, where we extract features from the key value pairs, which consists of some subset of the n-grams and ontology terms that we mapped from the first task, and we train a classifier to predict sample type. Here is the row normalized confusion matrix, where element ij represents the fraction of samples of type I that are predicted to be of type J. So you want to have high numbers along the diagonal, which you can see. And the overall accuracy that we have achieved so far is 0.84. And we've run all of these methods on over 150,000 human RNA-seq samples, which are available to query and download at metasra.biostat.wisc.edu. And also, if you have any, if you want to dig further into the methods that we've devised, you can look at the paper, which is at Bioinformatics. So just to show a couple features of the website, there were a number of features that we implemented to facilitate querying the sequence read archive. One of these features is an autocomplete feature, such that when a user is typing a query, we suggest ontology terms that we think they're searching for. So we require that users search via ontology terms. One challenge that we found, however, is that a user might not know about what terms are in the ontology. And so we're attempting to show the user what ontology terms are in the ontology via this autocomplete feature. And each of these suggestions is associated with both ancestral terms and descendant terms in the ontology to provide the user with information on what more general terms they can search for, as well as what more specific terms they could search for. We also show the results in a concise yet informative format, where we show the raw metadata, the raw key value pairs, as well as the extracted ontology terms in those colored rectangles where they're colored by the ontology that they come from. And to conclude, I'll just describe some work that's ongoing so this work focused on the metadata that describes the biology of the samples. We're now focusing on standardizing the metadata that describes the experiments that were performed on the data downstream of the living cells, such as what RNA-seq protocol was used, as well as what protocols were used to extract the RNA and so forth, and how their cells were grown. And we're using the MetaSRA for a cell type classification project where we're using the RNA-seq data in the SRA to train machine learning classifiers that will predict what cell types pertain to that sample by classifying the sample according to what cell types in the cell ontology we think belong to that sample. So this is a hierarchical supervised classification task, and the MetaSRA has enabled this project by providing the training labels. And with that, I'd like to thank my advisor, Colin Dewey, Professor Anhai Duan, Matt Ziegler, who is our lead developer in building the website. And for funding, I'd like to thank the Computation and Informatics in Biology and Medicine Training Program, as well as the Chan Zuckerberg Initiative. And with that, I'm happy to take any questions. Thanks. We map samples with terms from multiple ontologies. Yeah.
How do you deal with the, so there's probably some redundancy with different ontologies, and you try to merge them together, and try to mix and match some of the edges from
Right, yep. Yeah, so in regards to redundancy between ontologies, yeah, that is one issue, especially the experimental factor ontology borrows terms from other ontologies, but then they diverge. So a term that the experimental factor ontology borrows has diverged from the term in the original ontology. So part of that those procedures that we developed, one of those stages like links the ontologies. So we do try to address that problem, but yeah, improving this task will definitely involve trying to integrate these ontologies better. Yeah, so there are these thesauruses that kind of group them all together. Yeah, we haven't used that information yet. Yeah, we've used the raw information in the ontologies. Yep. Thanks.